Okay, so greetings everyone. Good afternoon, good morning, good evening, depending on which part of the world you are listening this from. And welcome to the 37th session of the online Optom learning series. Let me introduce to you to our speaker for today. Today we have Dr. Arathy Kartha, and she has finished her bachelor's and MPhil from the Shankar Nethralaya, Chennai. She then moved ahead to pursue her PhD in pediatric low vision rehabilitation from the Queensland University Technology, Australia. During her PhD, she has worked very closely about prolonged reading performance and visual functions in children with low vision. Currently, she is pursuing her postdoctoral fellow at the Welmer Eye Institute at Johns Hopkins University, US. And for her postdoc, she is working on developing methods to measure functional vision in people with severe visual impairment. And today, Dr. Kartha is going to talk to us about recent advancements in the sight restoration therapies. So we welcome you, Dr. Kartha, and we are waiting eagerly for your session.
All right. Thank you, Fakhruddin, for the introduction. Nice to meet you all here. I hope everybody is staying safe and healthy and remain like that. So today I will be talking about sight restoration therapies. There is no size that fits all. Okay, so let me give a little bit of introduction where I'm coming from. So my background is in low vision and low vision rehabilitation. So when you work with people who have low vision, very often they are being told that there is no treatment, and actually very frequently that there is nothing that can be done. So there are still problems in this universe for which there are no answers. That is, nothing can be done. So from today's talk, what I want to make clear is that there are so many things that is happening in terms of research, and there has been so many advances in the last few decades that it's very good to educate yourself about what is happening currently, which will really help you in patient education, and keep them motivated even with your low vision rehabilitation. So maybe like some of the conditions that they have, there is still not much that can be done in terms of treatment, but it is good to educate them about what is going on, and it's important to have the right information for that purpose. And so let me begin the session. So very often when you see low vision patients, they are very keen to know if there is any treatment. Even though they are here for the low vision rehabilitation, they want to know if there is a cure. So that is where the session comes from. Okay, so talking about blindness and visual impairment, there are about 1.2 billion people with uncorrectable visual impairment, according to the World Health Organization, of which 1/3 of them live in India. So we have a huge visually impaired population that we have to deal with. So before I begin, I want to give a little bit of background about clinical trials in general, because whatever I'm talking about, these treatments, they are all based on results from clinical trials. And you might be hearing this term so much more frequently these days since COVID-19 has happened, because whenever we are talking about treatments, we are talking about clinical trials. Any vaccine or any kind of therapy that are being developed, we hear the term clinical trial. Okay, we have to establish the efficacy through a clinical trial. Only then it will be available to the public. So it's a common term now, and it's important to understand what goes into it and the different phases that it comes with. So these are the different phases of clinical trials in general, phase 1 to phase 4. So how it differs is mainly with the number of people that are involved and also the duration of the study. So phase 1 obviously has the smallest number of participants, about 20 to 80 participants, and it takes about several months to do that study. And it's mainly to see the safety of a medication. So whether, that dose that you give is safe or not, if it has any adverse events like, big side effects or not. And then 70% of those phase 1 trials usually moves on to phase 2, usually. And in phase 2, it involves about 100 to 300 participants, so a little more than phase 1. And it takes up to 2 years to complete a phase 2 study. So it's slightly longer, as I said. This is mainly to see the efficacy. So now that in phase 1, you have established that the drug or the device or whatever you're investigating, it's safe. Now we have to see how effective it is. So, whether it is doing what it's supposed to do without, creating much problems like benefit to risk ratio and things like that. And then it moves to phase 3. So about 33% of phase 2 trials move on to phase 3. So you can see that, the percentage is getting less and less. So it's more, we are cutting off things and we are filtering it. So phase 3 involves about, more than 1,000 participants basically, and it takes longer, 1 to 4 years to establish this. And it affects the safety, efficacy, and also the dosing. So this is where they try different doses of a drug and to see which is the effective dose, basically, and what is the maximum dose that we can give with minimum adverse events. So that is called the effective dose. And it also, at this level, it also compares the new treatment that you're developing to the existing treatment. So if there is any current standard of care that is being followed, it's important to compare how effective the treatment that you're bringing in is better than the current one. Otherwise, there is no point introducing a new drug, right? So you have to establish there is some benefit over the current one to move on with this one. And usually at this stage, you might have heard about randomized clinical trials, which is also double-blinded, and it is masked. So both the experimenters and the patients are masked about the type of treatment that they get. So usually randomization happens independently without the knowledge of these two arms, the patients and the experimenters. And they are unmasked only at the end of the study to keep it like totally non-biased. So that is the idea of it. And when a treatment goes through a randomized clinical trial and then you can establish an efficacy, so that has gone through very stringent criteria to bring out the effectiveness of the drug. So after a randomized clinical trial is when we can clearly say that, okay, this dose at this level and this particular drug is clearly more effective than what is available. So that is a very stringent process. And once it goes through that, then you can actually release it to many, many participants. And that's a phase 4. It's then from then on, it is definitely, more available at that point. And then at that level, it is 70 to 90% success rate that it is going to be available commercially because it has gone through that process. So just to let you know, these are the different phases, because I will be mentioning this as I go through different slides today. So you should know what it is. All right. So just to give an overview of what I'm going to talk about today. So there are 3 things that mainly I will be focusing on. The first one is visual prosthesis. I'm talking about it because that is an area where a lot of research has happened and human trials have been done. And we have some, results which are very well established. So that is something which we know more than the other 2 areas that I'm going to talk about, which is gene therapy and then finally stem cell therapy. So this is in the order of how much information we have about these approaches currently. So let me go over to the visual prosthesis first. So if you look at this map, these are the different groups that are working on different types of visual prosthesis all over the world. I don't know if you are surprised. When I looked at this map, I was really surprised to know that these many groups are working on visual prosthesis. That is not one approach, different approaches, all with the same purpose. So there are about close to 50 groups all over the world working towards visual prosthesis internationally. Okay, so just to go back to a little bit of basic information about how the eye functions before I get into prosthesis and artificial vision. So this is something we're all familiar with as an optometrist, how the eye functions. Light enters the eye through the pupil, it falls on the retina, and then we all know retina has 10 layers. And the retina is wired backwards. We know that because the photoreceptors are way at the back. The light has to pass through several layers of neural tissue before it reaches the photoreceptors. We still don't know the exact reason why it's wired this way, why the photoreceptors are not up front instead of being at the back. We still don't know about that clearly. But after falling into, after it reaches the photoreceptors, it is processed, and then it passes through the neural tissue and finally to the ganglion cells, where it is transmitted through the optic nerve to the brain. So that is the basic function. So now, when we talk about different diseases, we know that there is some part of the system that is not working the way it's supposed to work. And clearly, we know that it is good if we can identify one disease which is much simpler to approach than doing everything. Like, it can be the different areas that can be affected, but if it is one layer or we know the area that is being affected, it is easy to find a treatment. So in this regard, retinitis pigmentosa has received a lot of attention because we know in RP, it is the photoreceptor layer that is affected, and pretty much the rest of the retina is intact. So we have an isolated problem there, and it is a much more contained issue to deal with. So because of this, most of the visual processes groups have focused on RP, basically, per se, and they have tried to replace the loss of photoreceptors. That is the idea. So let me get into the details. So there are different types of processes, but one that has been established and studied widely is the retinal prosthesis, because the retina is much more manageable, and we know much more about retinal processing than the other parts of the visual system. So if you go into higher order and much deeper into the visual pathway, there is very less information that we know currently about perception and processing compared to retina. So it's much more within reach, in fact. So in the retinal prosthesis itself, there are three kinds. One is an epiretinal prosthesis, the other one is subretinal, and then suprachoroidal. So epiretinal, as you can guess by the term, it is on top of the retina. So it is a surface electrode. It sits on top. And this picture here shows how it is. So the idea is that it is close to the retinal ganglion cells. And the idea is to stimulate the retinal ganglion cells that are surviving in retinitis pigmentosa. And the other kind is a subretinal implant. So it goes beneath the retina, and then it's aiming to stimulate the cells from the back. And the main targets here would be bipolar cells. And the third kind is a suprachoroidal, which is even more deeper, and it sits on top of the choroid. So the picture on the left here shows a little bit more detail from another view. So you can see the different kinds. These are the three approaches. So what I'm going to talk about is first about the epiretinal implant that we all might have heard about. It is the Argus 2 retinal prosthesis that is developed by Second Sight Medical Products in California. So this is a prosthesis that was the first FDA-approved therapy, visual prosthesis that was available commercially. And what it has is, you can see here, the patient is wearing a pair of glasses that looks like sunglasses, or you can have clear lenses as well. And it has a camera in the center, and then it has a coil here on the side, and it has a wire that runs, and it connects to a visual processing unit, which is a portable computer, basically. And then it has a coil at the back, which is surgically implanted. You can see here, and the electrodes are implanted on top of the retina. So it is similar to a surgery like a scleral buckling surgery, which we all are familiar about. So there is a band around the eye, and the coil is implanted on top of the fovea, which is the area where you have the maximum density of ganglion cells. So that is the idea. So how it works is that, like I said, so the camera functions as a video capturing unit. So it captures the scene, and then it is transmitted to the video processing unit, and the video processing unit processes the information. So every detail, like how you see in normal vision, has a lot of pixels, and you cannot transmit all that information into an implant like this because it is a lot, and that much of detail cannot be processed either. So it has to be downsampled. It has to be made simpler. So that processing or video processing is done in this portable computer, and it is simplified and then transmitted to the coil that you see on the side. This is a telemetry coil, which transmits the information wirelessly to the back of the eye, where there is another coil. And then it is transmitted to the electrodes, where it is electrically, the electrical signals are passed on to the retinal ganglion cells, which is pretty much like from there on, it is how the normal visual processing happens. Then the ganglion cells will take it to the optic nerve and then to the brain. So that is the idea. So it has many, many components that you can see here. And the surgery for this takes about 2 to 4 hours. It has become much simpler over a period of time with more people getting this implant. And currently, there are about 300, close to 300 people worldwide who has Argus 2 retinal prosthesis. So just to give you an idea about it. So this is how it looks like inside the eye. So you can see the electrode it's a 6x10 array, which means that it has 10 electrodes horizontally and 6 electrodes vertically. So altogether 60 electrodes. And if you compare this to how the normal eye is, you can see the scale of this attempt basically. So we know that in our normal eye, there are about 120 million rods. There are 6.5 million cones and more than 10 million bipolar cells. Okay, so that is the scale. And then once we lose these cells, we are trying to replace the lost cells with now 60 electrodes. So you can compare how it scales to, like, where is 120 million and where is 60, so there are some limitations here into what we can achieve with a prosthesis. It is nothing compared to the normal eye. We are not trying to even get close to that. I mean, it's still impossible, but someday I'm sure. But this itself is huge milestone to what we have achieved so far, because it involves finding the right materials that you can put in the eye that can last long, because it is sitting in a like surrounded by water or liquid, so it can't corrode, it can't make any reactions in the body. So there are several things that goes into it. So to find things that matches, and to find a camera that is miniature and then that can fit into those glasses, and the technical details, it is immense. So to even come this far is quite an achievement. But it's important to think about how the normal eye is and how this is as well, to put things in perspective. So this is a long shot to see how it sits in the eye. So you can see the optic nerve here, and then the fovea, and the implant sits right on top of the fovea. Sorry, let me go back there one more time. So a lot of factors goes into this. So even though, surgically, we try the best to get maximum closeness to the tissues, it's always not possible to get it completely aligned because the eye is curved, and this particular, the coil, sorry, not the coil, the implant is flat. So it is difficult to really follow the curvature of the eye. So it's not completely positioned at all times. It's difficult to achieve that. So there will be some areas which are less in contact compared to the other areas, which also impacts the performance. So these are things that goes into it as well, because of the shape of the eye. Okay, so the second one that I'm going to talk about is another type of implant, which is called the Alpha IMS. So this is developed in Germany by a company called Retina Implant. And this is another device which had approval in Europe, commercial approval in Europe. It's slightly different in that this uses a micro photodiode array. So the camera is integrated into the system here. So the person is not wearing an external camera. And also it has about 1,500 electrodes compared to the 60 electrodes that I was talking about in Argus. And this is a subretinal implant, which means that it is targeted to stimulate the bipolar cells. And it's slightly different from the Argus or the epiretinal implant in the way that because of the coil and the way the surgery is done, in patients with Argus, eye movements are an issue because they cannot make big eye movements. It's not possible because it can affect the position of the implant. So there is some restriction in eye movements there. Whereas in this case, because of the way this is made, it has better possibility of moving the eye, like natural eye movements are possible, because in this, the camera is within the eye itself. So it allows for natural eye movements. Whereas in the Argus, because the camera is external, moving the eye is not making any difference because it is a camera that needs to focus on the area that you need to look at. So you have to turn your head, and then that takes quite a bit of practice as well. So there is a difference there. So comparing like visual acuities that are achievable with these implants, with the Alpha IMS, the visual acuity recorded, the best acuity recorded was 20 by 546, ETDRS, not ETDRS, but roughly estimated. And they could read letters and they could have better visual functions. And this is the best achieved visual acuity among all the implants that we have so far. And it also, like I said, it achieved the CE mark in Europe, which means like they had permission to market it commercially. And both these implants, the Argus 2 and Alpha IMS, has since stopped commercial production since 2019 because the insurance and other financial issues, they couldn't market it to a larger population. It was really limited to certain population, and then the insurance coverage, and this is not a very cheap option. So it's a very expensive device, and then when the insurance companies are not covering the cost, it's very difficult to market it. So things like that, because of several issues, these two devices have since stopped commercial production. So another one that is being developed by a group in Stanford is the photovoltaic retinal prosthesis. So this is, again, a slightly different approach where photovoltaic cells are implanted subretinally. And you can see here the glasses have a projection screen, a tiny projection screen behind this, which is activated by infrared. And then the implant at the back is activated by the display here, and it's converted into electrical signals. And the aim is, again, subretinal, which means bipolar cells, to stimulate the bipolar cells. And currently, this is being investigated for AMD. So this is slightly different. And in AMD, in cases with geographic atrophy, it is the RPE that is being affected, and they have intact peripheral vision. So this is indicated for the AMD population, with severe visual impairment. And they are currently undergoing feasibility study in the US and France. So we still are looking forward to the results, but so far the results have been good. So this is another approach as well. So now coming to think about prosthetic vision, like I said, it is artificial vision, which is very different from native vision. That is something that you need to understand about this. So what the patients perceive is in the form of phosphene. So I don't know if you have experienced phosphenes anytime. You can experience it if you press your eye really hard, where you can see bright flashes in the eyes. Or if you bump your head really hard, that is when you get some phosphenes. So these are visual percepts, which is nothing like our natural vision. So when they see in the form of phosphenes, in the case of Argus, it is 60 pixels. So the vision is very different. So it is pixelated. And the functional outcomes are variable based on the behavioral adaptation, like how well you can get adapted to this kind of a percept. So it requires extensive training, and it varies between people. So it also depends on the surgical apposition of the implant. So how close it is to the tissues, and how well the implant is sitting in the eye. So it depends on that, the distance from the tissues and factors like that. And also it depends on the changes in the eye. So when a person goes blind and remains blind for a long period of time, the retina itself undergoes reorganization. So, the connections are different. There are abnormal connections between different layers. It is not as well organized as a normal eye. So the outcomes that you get when you have aberrant connections like that, it is very different from how it would be expected in a normal eye. So those things come into play when you consider the final outcome. But what we know currently is that from the functional outcomes, that they are able to, like, the functional outcomes vary between light perception. So the worst situation is that they're able to just perceive light and dark, to a very highly functioning group of patients who are able to read very large letters. And they're also able to perceive motion. And what we know that it works very well in high contrast environment. So which is not very common in the real world, but when we test them in control situations, what we found is that when the contrast is suppose like you're looking at a white object in a black background, then it can pick up much better. It works really well. So what we are trying to now do is to provide an environment like that. So using different type of filters. So instead of using a regular video camera, now we are trying to use image processing. So to convert the regular natural scenes into high contrast high contrast images, and then send it to the implant so that it's much more decluttered and it's much more easy to process. So that is currently what is being done. And I was involved in working with two of these filters where we used a thermal filter. Everybody is now familiar with the thermal camera, which is now used at all the airports and regular places to screen you for fever and things like that. So that is technology which is used widely now. So similar to that, the thermal filter uses an infrared camera. And so it filters out objects which are warmer. So if you're looking at a person versus like a non-living thing, it can actually pick up the person. So the person wearing this camera will be able to pick up humans or warm objects, or if they're looking at a warm cup of coffee on the table, then that will be highlighted with that camera compared to all the other objects around. So it actually filters the information for them. So it is much more easy to process. So that's one approach. The other one is to actually filter things based on depth. So instead of presenting them with the entire scene, in real world, it's often cluttered. Like when you look at an object, you don't just see that object. You have many things in the background which can interfere with your perception. So that can be filtered based on distance. So suppose you can set this camera for picking up things only at a certain distance, and by filtering out everything in the background, you can actually declutter the scene. So that's another approach that we are studying as well, which works well. So just to give you an idea about that. So now I want you to look at this picture and guess whom you're looking at. Do you have any idea what you're looking at here? You can type your answers and make your wildest guess. I just want to give you an idea about how pixelated vision would look like and what you can do with it. Like, just to give you an idea about what you're getting into. So by looking at this, can you say what you're looking at? Any guesses?
So I will help you with the answers.
Sure.
Few people are saying that a lady, a film actor, Abraham Lincoln.
Okay,
Face.
that's all very interesting. So let me actually go one level up then and see what they see then. Okay, so this is about close to 50 to 60 pixels that you're looking at, which is very close to the Argus limit. Okay, so when you look at a picture with the Argus camera, I mean, you don't see colors. Obviously, it's black and white in the Argus system. But, this is the amount of information, or much less than this is what you get. Okay, so any guesses now when you're looking at this? This is close to 500 to 1,000 pixels. Any guesses now? Are you able to get a better idea?
Yeah, two people are saying, I think 3 or 4 are saying Dr. APJ Abdul Kalam,
Wow,
lawyer.
that's amazing. Okay, so let me go further then. It's getting more and more better, right? So this is close to 5,000 pixels, and this is again getting there. And this is the picture that we are looking at. It has more than 100,000 pixels. Sorry, more than 10,000 pixels. So you can see the level of perception that you can get with different amount of pixels that you have. So now when I talked about the implant with 60 pixels versus 1,500 pixels versus, the other implants that are being developed, so you know how it can make a difference. So definitely, you would think that you won't be able to do much with less pixels, but you can see that you don't need much to do this either, because I could see that with the second picture, somebody was able to guess, so I don't know if you had guessed that even before, you thought about this, but, the amount of information we need to process things is much less than what you think. Okay, you don't need like 100,000 pixels to actually understand or recognize something. So it's very modest, so it's achievable what we can get with this. But with 60 pixels, there are certain limitations. Definitely, recognition can be difficult. Um, it might need like a lot of training, but with close to 1,000 or 5,000 pixels, clearly you can get close to normal functional vision. Like you can use it for navigation and things like that. You might not be able to read the newspaper print yet with that vision, but it's quite useful. You can see that. So next I want to show you another one. This is clearly what I'm showing you here is a bright window in a dark room. So if I ask anybody to point to this bright window in the dark room, I'm sure like everybody is able to do this. So even with this degraded amount of vision, this is also close to 60 pixels, but then this task is clearly very information. So there are some tasks like orientation that they can clearly perform that you can see now. Even though recognition, object recognition or face recognition would be difficult. I mean, that those are extremely difficult tasks even for people with normal vision anyways. So, apart from that, prosthetic vision can be used definitely for orientation and, high contrast situations like I'm showing here. So this is the actual picture. So it's not very different between these two. So just to give you an idea about what you can achieve. So now the second type that I'm talking about here is the cortical prosthesis. Obviously, we have been talking about the retinal prosthesis so far. That has been, undergoing human trials. But the other part of it is the cortical prosthesis. So retinal prosthesis uh, requires an intact optic nerve because you are conveying information to the ganglion cells or the bipolar cells. The expectation is that the information gets transmitted through the optic nerve and then to the brain. So what happens in those conditions where the optic nerve is impacted, like the optic nerve is the problem, like glaucoma or where there is a optic atrophy and where there is optic nerve damage. So for those conditions, you cannot use the retinal prosthesis because it won't be effective. So it is for those conditions that we need to think about something that can be directly put in the brain so that it can bypass all the other parts of the visual system. So a cortical prosthesis, in fact, was the first type of prosthesis that was developed even before the retinal prosthesis. So it was like in the early 1960s. It was first tried in humans by Brindley and Levin. And they had this system which was like filling up an entire room. It was very bulky, but then nevertheless, they found that it can actually give percepts to a person. So that was the most important finding there. It can induce phosphenes. So that in itself was a milestone. And then Currently, there are different groups that are working on cortical prosthesis. Even the second side medical products who developed Argus, they have a cortical prosthesis that is currently under investigation that is called the Orion. So it is very similar to Argus in the sense that it is a surface electrode which is implanted into the visual cortex, and it is directly stimulating the brain. And it is undergoing clinical trial currently. Another type of prosthesis that is being developed is the intracortical visual prosthesis, where the electrodes are penetrating electrodes. So it has more specific, like it can give rise to, the idea is that it can give rise to more refined phosphenes than a surface electrode. So that is being developed by a group in Chicago, and it is going into clinical trial pretty soon. But we don't know the results of these yet. From whatever we have seen from Orion, I think at least 5 to 6 patients have undergone implantation with Orion. The results are very similar to what we have with Argus. So they are able to perceive phosphenes, and their functional vision is very similar in terms of their acuity, like orientation or a direction of motion and things like that. So that is what we know so far. So it has wider applications for sure. It has more market than just people with retinitis pigmentosa. So it covers more conditions. So if this is successful, then a lot more people can get benefit out of this. Okay, so I'm moving on to the next part of this, which is gene therapy, which I was planning to talk today. So most of this comes from the idea that we know that most of the inherited retinal conditions are genetic, congenital. And so if we know the specific gene that is associated with a problem, then it is easier to, it is not easy, but at least we have some direction as to what to address. So that is the idea here, where after identifying a faulty gene, then the idea is to replace the faulty or the missing genes with functional genes. So they have identified about more than 200, close to 260 different types of genetic defects so far. So it is impossible to find one particular therapy that can fit into all those defects. So it is, we have to do it one by one. So I'm going to go over certain treatments that they have tried in humans so far, which are for specific conditions. So what we try to do here is to make working or functional copies of missing or faulty genes. First, that is the first step, obviously. And then we need something to carry those genes into the eye. So we need a carrier, which is also called a vector. So these genes are placed inside a vector, and currently what is being tried are the viral vectors. So they're using a virus to actually transport the gene to the eye. And virus offers, a virus is an ideal carrier or used for transport because of its natural ability to deliver the genes. Because you know how a virus enters the body. When it enters a cell, it replaces the genes of that cell with its own genes, and then that is how it alters the cells. So even like COVID-19, when you heard about it, you know that how it affects the lungs is it replaces the healthy lung cells with the faulty ones. So that is how it causes the lung pulmonary failure. So similar to that in the eye too, it has an advantage here when it is used as a vector because it has a natural ability to replace the genes of the cell that it is attacking with its own. So we are using that to as a tool for therapy. That is what is happening here in viral vectors. So there are different types of vectors that we can use. Of this, I will be talking about different types of vectors throughout this, different types of virus. So the first FDA-approved therapy so far, gene therapy, is Luxturna, which is a treatment for the condition Leber congenital amaurosis. It starts from birth. It's a congenital condition, and it affects children. The prevalence is about 1 to 100,000. And by the adolescents, they lose most of their vision. So it is very devastating, even though the number of people, it's very rare, but because when it affects children, you know the number of years that they live with the disability is more. So visually impaired years, if you count that, it is not less compared to an adult onset problem. So if you account for that, so it is a pretty devastating one. So it is good that this approach is working for children with Lebers. Why they were able to do this is because they could identify the gene that is causing the mutation. So it is the RPE65 mutation that is causing this condition. That is most common. So the treatment is specifically designed for an RPE65 mutation. So if it is an LCA with a different type of mutation, this is not going to work. So this is something that I will be emphasizing again and again and again, because even though the condition, the diagnosis is the same, if it is a different type of mutation, it is not eligible for that particular treatment. So in this case, it has to be a biallelic RPE65 mutation, which means that both the copies of the gene, like both the parents, have to give a mutant copy of the gene to the child to have to be effective, for this treatment to be effective. So you can see here, it is injected into the eye. So it's a subretinal injection, and this injection is developed by Spark Pharmaceuticals, and the name of the drug is Luxturna. It has that's a trade name. So coming back to the different type of vectors here again, so there are different types of virus that you can use, retrovirus, adenovirus, or adeno-associated virus. So currently, the most ideal one that they have isolated is the adeno-associated virus. It is suitable because it does not elicit disease. So when we use a virus, it's important that it doesn't bring out any unwanted effects or side effects. It should not be causing infection. And also it has to be targeting specific types of cells or tissues. So that is another part. So we have to, one thing is to isolate the gene, but it's also important to isolate the correct cell which needs the treatment. So it has to be very specific. And because of these two abilities, adeno-associated viral vectors have been very successful in doing this, and it has been ideal for gene therapy delivery. And this particular gene can carry up to, so when you talk about the capacity here, it's called kilobase pairs. So it means the amount of genetic information that this virus can carry, and this number is based on that. 4.7 kb is the amount that this adeno-associated viral vectors can carry. So it's the strength of, it's the length of the string, or DNA, the genetic material, the string size. Because I'm talking about this because we will talk about another condition where this is a problem. So that is why I'm mentioning this here. So you can see here, this is from the Luxturna, the website of the Spark Pharmaceuticals, how they have shown how this virus works. So once the adeno-associated virus is in the cell, it downloads a functional copy of the gene, and it replaces the faulty one. So to test this approach, one way, eye is a perfect organ because it is confined, it is contained, it's a closed space basically, which can cause very less leakage compared to other parts of the body, because there are gene therapies going on for very different conditions. There are different liver, different organs that gene therapy is being investigated, but eye is sort of a perfect organ for this because of this factor, because it's a small size, it's a contained space, and then so it's easy to target the cells that we want to. So it has been very successful, and it has chances of maximum effect. So Luxturna, how it works is that it's a single dose injection because you're altering the genes, you're altering the DNA. So they do it one eye at a time, and obviously genetic testing has to be done to confirm the diagnosis, and they have to confirm that it is the RPE65 mutation before they venture into this. So far in the clinical trial, we have the results from 31 patients who underwent this treatment. They had the visual acuity less than or equal to 20/60 and visual fields less than or equal to 20 degrees. And the participants were 3 years or older, so they had very young children who participated in this trial as well. So what happens is that when we have the functional gene, the function of RPE65 protein in the eye is to aid, is to help in the conversion. So in the visual cycle, if you look at the visual cycle, there are different stages. So the function of, you need to know the function of that specific protein to understand how this works. So what it does is that it helps in the conversion of all trans retinal esters into 11 cis retinol. So that is the part that RPE65 protein plays in normal eye. And when it is defective or when there is a mutation, it is this part of the visual cycle that is interrupted, and then they have this condition, the disease. So when we actually replace the gene with a functional RPE65, this visual cycle is reestablished, and that is what you see. So in these patients, when they did testing, they found that the visual cycle was improved. And also the other aspect that they found improvement was their navigation. So they did a test with an obstacle course. This was also developed by the company specifically for this. It's not a normal test that is available out there. So what they found was they could navigate it under low lighting levels. So before the treatment, or compared to people who have not received treatment, the patients who received the treatment could actually navigate the obstacle course much better and accurately, even under low luminance levels. So that is a difference that they found. And one more, so when we talked about different phases in the clinical trials initially, when I was talking about it, I told you the different phases examines effects over longer periods of time. So compared to phase 1, phase 3 is different because it is actually investigating the effect over a longer period of time. So it's important to understand that there are immediate effects, like obviously, the treatment has to work. That is important to know. But it is equally important to know if it is sustained. So how it goes over a longer period of time, if it's still maintained over a longer period. So when they did that study, now that the patients have completed many years after the treatment, so what they found was that the effect was maintained up to at least 3 years. But what they found was that the rate of loss of photoreceptors was same in the treated and the untreated retina. So that didn't change at all with treatment. And also, after 6 years of follow-up, then they showed that there was a progressive decline in visual sensitivity. So the effects was slowly starting to fade off after 6 years. So that is the problem here. Even though there are short-term effects, this treatment has issues in long-term maintenance. So that is something that they are trying to investigate currently. So the second one I'm going to talk about is another treatment for a different condition, which is for Stargardt disease. So this is called Stargen. It is a subretinal injection that is developed by another pharmaceutical company, which is Sanofi. And each of these drugs, when they are in trials, they have a code. They have a different name. Before it is marketed like Luxturna, or they are given names like trade names. They have a different code that they use. So that is what I have put in here in parentheses. So like I said, it is for Stargardt, and which has a which is more prevalent than LCA. It has a prevalence about 1 to 10,000, 1 in 10,000 people. And this is caused by one pathogenic mutant allele of ABCA4. So that is the gene that is associated with this condition, which is in the autosomal recessive form of Stargardt, which is more common. So this is a particular gene that is associated with that. So this particular treatment is targeting for this particular isolated gene. Like I said, so why I mentioned about the carrying capacity of adeno-associated virus initially was because when you talk about this particular treatment, the genetic material is much more bigger in size than the one that is involved in RPE65. So it needs a larger capacity virus to deliver this treatment into the eye. So an adeno-associated virus, AAV, is not able to carry this treatment. So they use something else called the lentivirus vector, which is a larger gene, up to 8 to 10 kilobase pair. So the other one I mentioned was 4.7. This is 8 to 10. So it can carry more genetic material, just to put it in simple terms. And so that this has been chosen for this particular treatment because of that reason. So currently this treatment was undergoing multi-center phase 1/2A study with 27 participants already recruited, but it was terminated in 2019. That was terminated not because of safety reasons. So phase 1/2A is mainly to evaluate safety and efficacy of the drug. That is the main idea in that phase, like I mentioned initially. So the study was terminated not because of safety reasons, but the sponsor, the Sanofi, decided to terminate. We don't know. So we don't know the results yet, but this was a clinical trial that was done. So probably like we can see the results later if they publish it. So in Stargardt disease, that it is caused by the accumulation of a product called lipofuscin, so which causes yellow flecks all over the retina. So another approach is mainly to target this particular issue. So we know that vitamin A is important for the eye. It is important for eye function, but in this case, what happens is that the metabolism of vitamin A is impacted because of ABCA4, the gene that I was talking about. The gene that is, when I talk about the gene ABCA4, it is actually the name of the protein that the gene is able to express. So ABCA4 is responsible for the proper metabolism of vitamin A in normal eye. And when that is impacted, when there is a mutation in that, vitamin A metabolism is impacted, and that causes more accumulation of lipofuscin. So that is a basic problem in Stargardt that causes this. So another approach is to target vitamin A metabolism, or to provide vitamin A without the accumulation of lipofuscin. So ALK001 is a synthetic vitamin A that is developed in the lab. It is a vitamin A analog that is developed by Alkes Pharmaceuticals. So they are trying to now give this vitamin A so that it can improve the sensitivity without the accumulation of lipofuscin. So that is the idea. And it has found to be effective in mouse models, which are in preclinical studies, in animal studies. And currently they are recruiting people with Stargardt for this phase 2 evaluation with ALK001, and also people with AMD, because the mechanism is the same in AMD as well. As there is lipofuscin accumulation in AMD as well. So there are two studies that is going on. In these two conditions with Stargardt and AMD. The AMD is a little bit more advanced. That is in phase 3 already. It is undergoing a randomized controlled trial currently in people with geographic atrophy. So when we can see what the results are, but the animal models are quite promising. So we have to wait and see. Next one is ushers. We are familiar with ushers because it is a certain type of RP where people are both deaf and blind. So it is a dual sensory impairment. And USH-STAT is a treatment that is being developed for Usher syndrome type 1, where there is a mutation in the protein myosin 7A, it is called myo7A, which causes problems in ear and the retina. So that's the reason behind the dual sensory impairment in this particular type of Ushers. And what they're trying to do is to give a subretinal injection for myosin 7A. And this is currently in phase 1/2A, where they're investigating different doses currently. So they have 2 doses that they are currently studying to see which is the effective dose. And the study was terminated early as well. So this is something that you have to keep in mind. There are clinical trials happening, and then they even though in animal models and other small studies it is effective, sometimes a decision has been made to terminate the study. Again, in this case, it was not for safety reasons, but they decided to terminate. So we don't know with the existing number of people what the effect was. So now it's slightly different. It's gene therapy, but it's different in the sense that it is not DNA, but it is RNA therapy. It is for Ushers again, but a different type. It is called Ushers type 2A. And this is developed by a company in Netherlands, which is called Procure. And this particular product is injected intravitreally. So it is not a subretinal injection, it's intravitreal injection. So subretinal injection is a little bit more complicated than the intravitreal, as you know. The procedure is much more invasive compared to an intravitreal injection. So in this, it's intravitreal injection, and this particular product is something called AON, that is the antisense oligonucleotide, which is RNA that masks the mutation basically. So it's not replacing or anything, it masks the mutation so that normal functioning is established. So that is ongoing currently. So this is an RNA therapy. Another similar RNA therapy that is being undertaken by the same company is for LCA10. So this is a different type of mutation compared to the other one that Luxturna deals with. As I mentioned, Luxturna is for RPE65, and this one is for another one called PCIS998X mutation in a different gene, CEP290 gene. So that is the mutation that this is working on. And this is in phase 1-2, and phase 2-3 is ongoing currently. So it requires multiple intravitreal injections. So unlike the DNA treatment, where it's a one-time subretinal injection, in this case, you need multiple injections. So because it doesn't cause any permanent change in your DNA, like when you do this. So you need repeated treatments. With RNA therapy, that is one thing that is different. But they have done the study over 12 months with multiple injections. What we have found is that there is at least 5 lines of improvement in vision, and also they have improved in sensitivity tests and mobility at 12 months follow-up. So that looks pretty promising for this particular condition, which is LCA10, again with this specific type of mutation. So we have to see the results of a larger study, but so far so good. Another type of treatment is using ciliary neurotrophic factor. It's also called CNTF. So broadly, neurotrophic factors are present in the body, and this particular one, CNTF, it's known to retard retinal degeneration and when they studied in animal models. So now they are trying to investigate this in humans. This is again tried in Ushers type 2 and 3, and also for retinitis pigmentosa, currently in phase 2. Which means it's again studying safety and efficacy at this level with close to less than 1,000 participants, basically, because it's still in phase 2. So this is an intravitreal implantation of an encapsulated module. So it is not a molecule that has been injected, but the product is actually encapsulated in a semi-permeable membrane, basically, because if you directly inject it, it will cause a reaction in the body. It can, cause inflammatory reaction or rejection. So to prevent that, it is actually put in a capsule and then intravitreally implanted. So this particular method actually allows for the neurotrophic factors to enter the cells. But at the same time, also the nutrients to enter the capsules. So, it's a semi-permeable membrane. And now they have been followed up to 30 months post-implantation, and we have results from 3 patients from 24 months follow-up. So what they showed was that there is the thickness of the outer retinal layers is better in people who received the treatment. Not people who received, so they used 2 eyes. So one was a control eye in the same patient, and one eye received the treatment. So they compared between the eyes. So in the eyes that received the treatment, they found that the retinal layers were thicker. which is promising because that shows that it prevents the retinal degeneration. And also the sham eyes showed more cone spacing. So the spacing between the cones was more in the eyes that do not receive the treatment. Also, there was a decrease in cone density compared to the treated eyes. So definitely the treatment is showing some effects in terms of how it changes retinal degeneration. So we are still waiting for the complete results of this, but so far it is good. Okay, so this is another condition which we are familiar with again. It's called the X-linked retinoschisis, where there is a splitting in the retinal layers between the neural and the sensory retina. And the prevalence is 1 in 5,000 to 1 in 20,000. So it is, again, not that rare. And in this particular protein affected is the retinoschisin, which is responsible for, obviously, for the adhesion of retinal cells. When it is impacted, it is less adhesive, and then there is splitting. So that is the pathophysiology behind it. And the study is now in phase 1/2, where they are studying different doses for safety. And once it's effective, so far they have seen promising results, and we are waiting for the results of this also. And this is another condition again, choroideremia, which is X-linked recessive, and it affects males more, where there is a progressive degeneration of photoreceptors. And they have, similar to RP, they have tunnel vision, that is peripheral vision loss, and it is progressive, and by adolescents, they lose most of their vision. And the particular mutation here you can see is the REP1 mutation for a specific protein that they could identify. And then they are now using adeno-associated vectors to replace this faulty mutation with a functional gene. And then they are studying with humans. So they have recruited people with vision less than or equal to 20/30. And they have shown that the visual acuity is better, and there is increased sensitivity when they measure the dark adaptation. And results are available for 3.5 years, and it shows that the eyes are stable compared to control eyes. Other studies are ongoing from other companies as well, like Spark and Biogen. So this is another condition where there is a lot of treatment options happening. Another one is achromatopsia. So this is an autosomal recessive condition. It affects 1 in 30,000 people. It is stable. It is congenital, but it is stable. It's not a progressive disease. It's mainly cone dysfunction, and there are 2 mutations that are identified in this. And now, with these mutations, they are getting subretinal injections. It's done by a company called AGTC, and it is ongoing. So currently, there is no other treatment for this. And so it's interesting to see what the results would be. So retinitis pigmentosa, as we know, it's a complex disease. It has at least 60 genes which are associated with just RP. So it's very difficult to find one promising treatment for RP because it's so variable. And this particular type of RP38, is very rare, and it's mainly found in isolated populations in Middle East and Faroe Islands. It's an autosomal recessive condition with a specific mutation in NERTK. That's what it's called, and it mainly causes disruption in RPE. So it's slightly different. And so by giving the subretinal injection, it has been found to be safe and effective in phase 1 that was done in Saudi Arabia in 6 people. And it's been maintained up to 2 years that we know. And then after that, they showed some decline in 2 out of the 6 participants. So it's definitely got short-term effects, but whether it's maintained long-term is something that we are yet to see. So now, so far, what I've been talking about is replacing the gene. So it's using a viral vector. You replace the faulty genes with a functional copy. So they are still trying to develop methods to find more effective ways of for injecting the gene in the eye. So one way that is currently being investigated is gene editing instead of gene replacement. It is gene editing. So you cut off the string that is defective and replace it with a functioning one. So that is the idea behind this. So it is still in very early stages. Currently, the study is being done for LCA10 that we just learned about, that was undergoing therapy with the RNA therapy. So it is the same condition that is being investigated for CRISPR. It's called CRISPR, which is a gene editing technology. And Allergan is doing a study for that. It's called EDIT 101, and the inclusion criteria, 3 years or older, people have received this, and it looks pretty promising as well. So this is another approach. Basically, it is also gene therapy, but it is a different approach. It's called a gene editing technique. So now what I'm going to talk about is something called optogenetics, which is gene therapy, but it is a different approach where you are making the remaining cells of the retina. So if in case of loss of photoreceptors, you lose the light sensitive cells. Photoreceptors are the light sensitive cells in the eye. So when you lose that, the remaining cells like bipolar cells and ganglion cells, they are finding a way to make that light sensitive. So by altering the genetic properties. So by injecting a specific protein called opsins, we know opsins, those are responsible for the light sensitivity. So by injecting opsins and making the existing cells light sensitive, they're trying to replace the lost photoreceptor. So that is the idea behind this, to make the remaining retinal cells behave like photoreceptors. It is indicated for RP currently, and it targets mainly bipolar cells and retinal ganglion cells, and it is still very much in the experimental stages. So once developed, it can be used independently or along with, visual prosthesis. So that is yet to be seen. So final part of my talk is on stem cell therapy. So this is very much in the early stages. The project that I'm going to talk about first is the California project to cure blindness for AMD. So people with non-neovascular AMD or dry AMD, they are trying to replace or compensate for loss of RPE. Basically, that is the mechanism in AMD. So the cells that they use are human embryonic stem cells, stem cell derived RPE. So HESC or human embryonic stem cells are cells that can be differentiated into any type of cell in the body. So these are present in early embryo, like it's called a blastocyst. So in that phase, in the human embryo, you have stem cells available that are capable. It's called pluripotent. So they're capable of differentiating into any type of cell in the body. They can be brain cell, they can be your spine, they can develop into anything. So they have the capability of doing it. And using the human embryonic stem cell to behave like RPE cells, they are able to implant it in the eye on a synthetic membrane. So the idea is a layer of human embryonic stem cell derived RPE on a synthetic membrane because it has to be biocompatible. In this case, for this study, they used something called Paralin. And then in a phase 1 trial for safety, they found that the visual acuity improved by 17 letters. And also another outcome variable that they assessed was better fixation. So the patients showed better fixation because in AMD, they have fixation instability. So when they studied this particular method, actually showed better fixations in people with AMD as well. So this is another approach where stem cell is used to develop RPE in the lab, and then it is transplanted into the eye, and then create a new layer of RPE in the eye to compensate for the RPE loss in AMD. So this is again done in people with very advanced disease like geographic atrophy, but it shows promising results so far. Another one is the London Project to Cure Blindness, a very similar attempt. It's in phase 1 study. Again, human embryonic stem cells on a synthetic membrane. So each of these approaches are investigating different types of membranes that can act as a biocompatible interface between the eye and the layer of cells, so that it is not rejected after implantation. So they are studying different membranes and different materials at this point. And then this particular one has also been successful in wet AMD. So the other one was dry AMD, and this is the first study to work with patients with wet AMD. And they have the results from 2 patients where there was improvement in visual acuity, 29 and 21 letters. And this study also checked reading speeds for these people. So we know that AMD causes central vision defect, and most of the people complain of reading problems. So one of the outcomes that they studied after this treatment was reading speeds over 12 months. And what they found was they had improved reading speeds. A different method. So we know that human embryonic stem cells, when they have been used in research, it was met with a lot of criticisms because of using embryonic cells. So there have always been the idea to look into other methods to create RPE cells. And one such method now being developed is called the patient-derived induced pluripotent stem cells, or iPSCs. So this was first developed by a lab in Japan, and now there is a study ongoing by the National Institute of Health in the US as well, where the cells are taken from the patient's own blood, and then they are converted into stem cells in the lab, pluripotent stem cells in the lab, and then to derive RPE. And then using a patch of cells implanted in the eye, they are studying improvement in vision. So because it's taken from the patient's own cell, there are less chances of rejection. Or another aspect of stem cell is that, if it is not very specific, it can cause tumors. So because it may not just target the cells that we intend to change, it can also target other cells and develop into tumors. So that's a problem with stem cell therapy. So because it's taken from patient own blood, there is less chance of rejection, and iPSCs are also known to have less tumorigenicity. So these are the 2 advantages of this particular treatment. There is also a stem cell therapy being developed for RP that is in the phase 2B stage by a company called J-Site. So what they use is something called human retinal progenitor cells, which is the cell from the retina itself. So we are using the cell taken from the organ that we are dealing with. So here it's from the retina. So this is clearly, the way it has to be if it works. So it has the potential to be a ideal treatment. It aims to replace lost photoreceptors as well as slow down progression. And this study is expected to complete by next year, early next year. So that's something we have to wait and see. So from all the different therapies that I discussed, it's very clear at this point that every mutation is different. Every different protein has a different function. So there is no one size that fits all. You cannot find one treatment that can cure everything. It's impossible, as it is clear. And so far, only about 260 genes, or more than 260 genes, have been identified with that is associated with inherited retinal degenerations, and there are still more that are being investigated. So and there needs to be a treatment for every specific mutation, so more genes may be identified in the future. And also, you have seen now that most of them have been targeting end-stage retinal degeneration. So whether it's AMD or RP, it's mostly for people with advanced disease. So when they restore vision, it is not that the vision is restored to 20/20, but most of them end up still with low vision. So even in the severe visual impairment range. So even visual prosthesis, it is intended for people who are blind. With RP, it actually provides restores vision to the ultra low vision range. That is what it is called. that is vision less than 20/1600. So that is the range that you get even with these treatments. So that is something that you have to keep in mind, that it does not restore vision to 20/20. And most of them, or all of them, still need extensive low vision rehabilitation to function or reach their maximum potential. So that is something when you educate patients that you have to keep in mind as well, that this is not going to be replacing low vision rehabilitation, or it is not going to restore 20/20 or normal vision. It is very different from normal vision, at least in visual prosthesis. It is completely different from their native vision. So there is still more work to be done in the low vision field, because we need to develop specific functional vision tests that can now test people with this range of vision. Because currently, when we use the visual acuity charts or the logMAR charts, even when you bring to the closest distance, it can only measure up to a certain range. It cannot do vision less than 20/1600, even at the closest distance. So how do you test vision in these people? Like you saw in the case of Luxturna, they have developed a mobility course to assess functional performance. There need to be more functional vision tests. So that's something that I've been working on for the last 1.5 to 2 years, is to develop tests for people with severe visual impairment, which can also be used for people who have undergone clinical trials similar to what I've been discussing. And the need for extensive low vision rehab cannot be stressed more. So that's something you have to keep in mind as well. So all this information that I shared today is available from this website. So most of the clinical trials, now it's mandatory actually to register on this website. It is present, it includes studies from all countries and all different areas. So you can actually visit this anytime to update yourself. So what I presented today is already outdated tomorrow because there's so much going on. The results keep changing, studies get terminated. So you have to keep yourself updated about this information. If you're interested, this is a site that you should visit, which gives you reliable, because it's also important as to where you get the information from. So I would recommend visiting this website, which is clinicaltrials.gov, which is a complete database for all clinical trials. And you can type the condition and you can set filters here, as you can see. So most of the studies that I discussed today can be found here as well. And thank you all for listening, and thank you for staying till the end. I really appreciate it, and I'm happy to answer any questions.
Okay, thank you very much. I think it was a very brief, and I think you very well did the justice to the topic, that which says that one size does not fit all, and we saw with different conditions you had different types of therapies and which are going on. So thank you very much for sharing the knowledge across. There are certain questions. I'll just read out a few for you. Somebody is interested to know what would be the youngest age at which Argus 2 prostheses have been implanted.
Okay, so that's a very interesting question, actually. So you know, Argus is being developed for people with retinitis pigmentosa, right? So retinitis pigmentosa is a case where you have progressive loss of vision. And so most of them have very good vision in their young age, and close to like mid 40s or early 40s is when they start to lose their vision. And after that, at different ages, they lose their complete vision. So only, if the vision is in the light perception or bare light perception range, Argus is indicated, because anything more than that, Argus is not effective, because the amount of vision you get with the prosthesis is very similar to the ultra low vision range. So if you're already ultra low vision, or if you have any little bit of amount of vision, natural vision left, this is not for you. So once you reach that stage, because it is RP, patients are usually in the late 50s or, very in the older range. You don't do it in young children because of the disease itself. But if you, now expand it to other conditions, the age groups can differ. So that's a completely different thing. But it's purely because it is RP and the natural progression of the disease that, the age group of the people we have is from 50 to, I have seen up to one of our patients is into the mid 80s or late 80s. So
Oh,
yeah,
okay.
yeah.
I see. And again, continuing to the same question about the Argus 2 prosthesis, something like how does the cord implant stays hanging out of the eye, because there was a cord which was implanted. So is it like a just like a transmission to the chip, or there is a hanging cord outside which attached to the glasses? So I think that was not clear to that attendee probably.
Sure. So, like I said, I mean that's also like it's very hard to imagine these things when you see the picture. But then, so the chip is the part that is in the eye, like on the fovea, and the rest of the area, the cable and everything is around the eye, and then it is stacked in the eye. So it's very similar to how it would look like in a scleral buckle. Like that is the closest that you can imagine. It's a retinal detachment surgery. You know how the buckle looks like, right, around the
Yeah,
eye. So
yeah,
it's
yeah.
very similar to that. And then,
Mhm.
rest of the things is wireless. So they have a coil in the eye that actually communicates with a coil that they have, on the temple that is attached to the glasses. That is the wireless communication. That is this has a radio frequency and it communicates
Mhm.
with the coil inside. So that is how it works. So there is no wire that is coming out, through the
Oh,
head and things
okay.
like that.
Yeah.
So if that makes sense, if that is what you
Yeah.
meant. Yeah.
Yeah, yeah. I think that's what was the question. I think the transmission happens to the coil which was on the glasses with the coil which is inside. So probably that's the thing I think which was to be answered. That's correct. And some people are asking any idea about all these types of prosthesis availability in India, or is it still in available only in the US? Is it available overseas? Any idea about that?
Sure. So that is a question which, I have always asked myself too, because, I mentioned at the beginning, it makes sense. One third of the population lives in India and we have a lot of RP. RP is very
Yes.
common in India. So ideally, like I would be like, why don't we take this to India? Like, you have the population there. So what I presented here, like you can see, the processes that got the FDA approval here was the Argus, and then that was covered in the US by certain insurance companies. Not
Mhm.
everything, but then people who had Medicare, which was the main one, for the elderly population. So the Medicare was able to cover it, which is why the patients could afford it. So whichever country, like even in Europe, when they had the market there, uh, they had a contract with the insurance companies where it is covered by the insurance. So you could have it, you could give it to people. In India, that
Mhm.
is the main problem. Then the affordability, unless like it becomes mainstream and it goes beyond phase 3 trials, like larger clinical trials are done, it's not available immediately, correct? So
Mhm.
currently, like with the minimal availability, if you bring it there, it most of the people won't be able to afford it. That's the thing.
Yeah, yeah,
And
yeah.
compared with the cost and the benefit, it is very hard to make the judgment. um,
Okay.
so eventually, like I do see that eventually, yes. I mean, maybe like who knows, in the next, decade, one of the groups in India itself will come up with something more affordable. You may never know.
Yeah,
And then, that would be the way to go with it. But then right
yeah,
now, it's still, there is a lot more that are unknown to us currently,
yeah,
because it's
yeah.
still in the research stages. But
Still in the
once
phase
it,
one, two,
yeah,
three. Yeah, maximum is
we
three.
have to, right, we have to refine a lot more. And also, like I said, even with the prosthesis, there is a lot of rehabilitation that has to go into it. So people who are, I mean, most of the people that undergo this already have undergone rehab and they're using a cane, and they have
Yeah,
all the other things available, like all the other sensory modalities available that they're using. Because with Argus or any other type of retinal prosthesis, it's not an independent thing. You can't just use that and go out in the open,
yeah,
because you
yeah,
need to use it along with other strategies. You still have to use your cane, you still have to use your other devices and gadgets to make you
yeah.
safe around with the device. So how far that is, because of the infrastructure in India, how far that is possible in India is a big question. To use
Mhm.
these devices and walk around. I mean, how safe it would be, with the infrastructure, that is also a question.
Suppression.
So we have to
Yeah,
see,
that's true as well.
yeah. So, I mean, same thing with magnifiers and telescopes as well. I mean, when you prescribe a telescope, like, you're always, warning people where to use it, like how can they use it, Mostly they can use it when they're stationary just to spot things and, things like that. But after that, it's not safe to walk around with it and things like that. So there are a whole lot of things that need to be considered, that you have to keep in mind, but definitely not impossible. I mean, in the coming
Yeah.
years, I do see it. I mean, that's the way to go. I do see that, because compared to all the other options, prosthesis is something that is more, like it has more advanced since the last few, years. The field is, moving very fast. So definitely we can check that. So keep checking on that website to see what is going on.
Yeah, yeah, and I think thank you for sharing that, because you have to keep yourself updated, as you mentioned, like what information you have shared with us today. There'll be some positive developments, or
Yes.
the research have been closed down or whatsoever. So I think it's now moving on to evidence-based practice, where you need to look at the evidence and what's happening in terms of trials. So thank you for
Yeah.
sharing that with us, of course.
And gene
And
therapy too, like, that might become much cheaper option compared to processes like in the coming years, because a lot of advancements are happening in terms of the vectors to use and, the specificity of the cells and treatment and everything. So that is really rapidly advancing as well. So, maybe that is the way to go, because that provides with more natural type of vision than
yeah.
processes. So, like, yeah.
So the again a question comes for cortical visual impairment (CVI) patients. So will all these work for them in terms of I think specific question is for cortical prosthesis. Any ideas whether it will work for CVI patients?
So the one thing that is required for cortical prosthesis is that the cortex should be intact, right? Because any other part along the pathway can be impacted, but you need an intact visual cortex, because it is a cortex that is being activated when you're using the cortical prosthesis. So because of that exact same reason, it may not be possible for CVI patients, like, because they have a cortical origin, like cortical problem. So currently, yeah, that
That's a gray
might
area
not be a possibility
probably.
for that particular group.
Yep.
Yes.
And what's your take on the visual prognosis after gene therapy? I mean, how's the visual prognosis? Does it depend on certain things or?
So right now, what we know is definitely from one particular treatment, which is Luxturna, that is the most, that is the most successful one. Basically, they have the FDA approval, and they have, quite a few participants with that, mostly children and young adults. So what we know is that definitely they have found improvement in the functions. In terms of vision, maybe not that much. Like when you put them to read on a visual acuity chart, no, I mean, you don't see like a huge improvement in their vision. But like I mentioned in the talk, like what they found is that the sensitivity is better, like the dark adaptation is better, because, like it is the visual cycle that it is, that it is addressing, basically, that is flipping, like reverting back to normal. So because of that, visual sensitivity is definitely better. And because of that, they can, the luminance range in which these people can operate is different before treatment and after treatment. They are able to function much better in low luminance and move around much better, like mobility is better and things like that. So functionally they do find some improvement, but quantification, like, that is why we need tests. Like we are still developing tests to address this, actually, to how to quantify the visual improvement. So that is another huge area that is being developed as well, right? But currently we know, yes, functionally they are better, but VA, like in terms of the number of lines or letters, not much.
That's right.
That is with
Yeah.
Luxturna. The other therapies are all still ongoing, and we don't have as much information as we have about Luxturna. So I can only say like a little bit of surety, like in terms of Luxturna compared to other treatments, because we still have to have more information.
Yeah. Yeah.
That is the only treatment which has gone into phase 3 currently. The other ones are all still in phase 1 or phase
Phase
2.
1, 2. That's
Yeah.
right. That's right. So I think keeping ourselves updated again. I mean, we are just reminding us to look at and keep updating ourselves to get better knowledge each day by day.
Yeah,
And we would
because
emphasize day by day because everything is changing every day now. So
yeah, I
yeah,
mean there is a lot going on. And also like these things are important because I remember like when we saw patients, sometimes, the more active, proactive patients, sometimes they come with these paper cuttings to you, like from the
yeah,
newspaper. Like there is treatment for this condition, like, and then I have this condition. Can I get this treatment? Things like that. So, what is shown in the media is sometimes very different from, the actual truth. Like, you have to scientifically back it up and you have to tell the correct information to this person. You can't tell like, no, I mean, you won't be able to get this treatment, or yes, I mean, give them false hopes that yes, I mean, it's going to happen. So it's a middle ground. Like you have to say, okay, this treatment is successful for this specific condition with this type of mutation,
that's
like
right.
it's,
That's
it's
right.
important to educate people the right way. So, and also for yourself, you have to understand, what it takes to develop something like this. Many of you,
Yeah.
if you're students, you might get into research and you might want to, be involved in one of these streams. And then, you don't know what excites you. So it's important to read up and keep yourself updated about what you want to do as well. So, yeah.
Yeah. Yeah, that was the whole idea behind when we were drafting or thinking about the talk, when you mentioned that you would be sharing all the information. And we were like, as an optometrist or as a primary eye care practitioner, we must be aware what's going on. As you said, people might just come up with a newspaper cutting or a website a screenshot saying that this is available.
Hmm.
And if you are not aware about this, it's a bit awkward in front of the
Yes.
patient, and you are
You
the
can't
one
give
who
false
are
information.
supposed to guide them properly. So I
Yes.
think it's really
It's your
important to keep ourselves
responsibility.
updated.
Yes, yes,
Yeah.
absolutely. And so, like each of the things that I covered today is a talk in itself. Like each of the slide, I can actually do it for 2 hours, like that single slide, There's
Yes,
more
yes.
information about it. I haven't gone into the details about, how each therapy, like subretinal injection, what exactly happens, or each gene, like those are like specific topics in itself, actually, that you can deal with. This is just the tip
Yes,
of the iceberg. That's what I wanted to say. What I dealt with
yeah.
today, there's so much happening that it's impossible to cover in one talk. So
That's
feel
right.
free
Yeah, yeah.
to reach out to me. Like if you're interested in any specific detail or anything, I can always help you get that to you. So you can always write to me. I have given my email up front. And also, like, if you want to know anything about visual prosthesis or anything that is happening in the lab that I work in, like with my mentor, his name is Jilan Danieli. So in our lab, you can always visit our lab website and, come back with more questions. So I have given both upfront in my first slide. So feel free to do that, because it's impossible to cover everything, in this short span of time. But
Yeah.
this is a great attempt. I really, like what you're doing here, with this series. I have taken time to watch your other talks and everything too. It's so informative and it's been great. So, yeah.
Thank you so much for taking out time and watching the series, and also for your wonderful talk. I think we have taken most of the questions, and we have also, I mean, you have covered a lot of the questions which were asked during your presentation. So I'm just skipping all of that because the session is already recorded and people can watch back the session. And a lot of positive responses. Thank you. And I'm reading out the chat. A lot of positive responses, and thank you for your informative talk, uh, which has been posted by our attendees. So thank you very much, Dr.
Thank
Kartha,
you.
for sharing the. I think it was not the tip of the iceberg. I think for us it was the whole iceberg, because most of us didn't even know about. I think 80% of the things what you talk about. So I think it's very good, and thank you so much for doing this.
Sure. Thank you very much. My pleasure. And keep yourself safe. And thank you for Krudeen. And, um,
Yeah,
take care. Have a
most
good
welcome.
weekend. Yeah.
Thank you very much. Tomorrow, we do have a session tomorrow, tomorrow evening, which will be late evening at my time. So I'm just going to share my screen. So yeah, we do have a session. We have Professor Bruce Moore again coming back, and he is going to talk to us about the refractive error management part 3, where he will cover more details about specific conditions like prescribing in amblyopia, strabismus, and he will also talk about the interrelationship between refractive error and those conditions. So please tune in tomorrow. The timings are 6:30 in the evening Indian time and 9 p.m. late evening Malaysian time. Until then, stay home, stay safe, and see you all again tomorrow. Bye.